Hi everyone! Okay, so we're going to be talking about the chromosomal basis of inheritance. The last chapter we were discussing genes and inheritance patterns, but now we're actually going to be talking about genes that are found on chromosomes. We're going to be discussing some genetic disorders that are chromosome based. So let's go ahead and start. So what's the relationship between genes and chromosomes? Well, genes are found on specific locations on chromosomes. So as you guys can see here, here is a drawing of a chromosome, and the loci, that word loci, is basically plural for location. And on this chromosome, you can see here that it's showing the location for the gene that's going to give us eye color. And then the phenotype is the color, okay? And then over here would be the loci or location for the hair color gene. Hazel. I don't think I've ever heard that term for a description of hair color, but oh well. So genes are found on specific loci or locations on the chromosome. That's the relationship between genes and chromosomes. What is the chromosome theory of inheritance? There's two parts of the theory. Number one, it states that genes are found on specific locations. We just stated that. Okay, so that's the very first part of the theory, is that genes are found on specific locations on chromosomes. And in fact, with very simple organisms and even recently humans, we have mapped out where genes are found on all of the chromosomes. Genes are found in specific locations on chromosomes is the first one. Number two, chromosomes undergo segregation and independent assortment. So by now, hopefully you understand what that is referring to. Remember, segregation refers to anaphase 1 of meiosis, where the homologous pairs are going to separate. So here is a pair, here's another pair, one from mom, one from dad, and the pairs are going to separate during anaphase 1 of meiosis 1. And then the independent assortment refers to metaphase 1 of meiosis, where they line up. If we have genes that are on separate chromosomes, then they're going to, independent of each other, they're going to assort independently into the gametes. Okay? So that was segregation, the law of segregation, and the law of independent assortment. And on the chromosomes, like I was saying before, on the chromosomes, we've basically mapped out all of the genes that are found on chromosomes. And don't forget that chromosomes, what are chromosomes? Chromosomes are basically DNA wrapped around proteins, tightly around proteins. And you can, we're going to be discussing how do we map out the genes that are found on chromosomes. So here is a homologous pair of chromosomes, and again, one is from mom, one's from dad, and so we receive two genes for every trait. So maybe over here, you would find, and over here, you would find the gene for eye color, and then maybe on this location of the chromosomes, you would find the gene for enzyme A, and then maybe over here at this location, you would find the gene for cytochrome C. So we have two of every gene, okay? I mean, here you can see the duplicated chromosome, and then so the sister chromatids are going to separate into the gametes, right? So like this sister chromatid, this sister chromatid, they're going to go into separate gametes. This sister chromatid, that sister chromatid, they're going to go into separate gametes. So you end up with 1, 2, 3, 4 gametes. And remember, gametes are haploid. They have one set of chromosomes, right? So you're either going to give away, remember the law of segregation, you're either giving away the chromosome with genes from mom, or you're giving chromosomes with genes from dad, okay? So Thomas Morgan, he was the first scientist to associate a gene, a specific gene, to a chromosome. And what he had discovered was that the gene for eye color, red versus white, okay, that white color is found on the X sex chromosome of fruit flies. Okay, and he also discovered that the red color is what he called the wild type. Okay, the wild type. And then the white eye color is the mutant, or the mutant type. And what he noticed was that when he found wide-eyed flies, oftentimes they were found on males. So he was able to do some experiments, and he discovered, he came to the conclusion that the gene for eye color, red, or the mutant version, white, is found on the X chromosome. Okay, so let's go ahead and write down the genotypes for the following males and females. What is the genotype for the wild type male? It's going to be X, big R, Y. So wild type has red eyes, which is dominant. Wild type females are going to have X, big R, X, big R. Wide-eyed mutant male, X little r, Y. Wide-eyed mutant female, X little r, X little r. Okay, so those are the genotypes, and then these are the phenotypes. And the reason why I wanted to write this out, because now we can take a look at his experiment. So the P generation, he had purebred red eyes, and you know the way that he symbolized wide-eyed versus red eyes is kind of strange. Okay, he did W plus, which means red, and then W, which means white. So you don't have to use the same lettering that he did. Like for me, on the previous slide, I just wrote X big R for red. And that makes sense to me because red is the dominant trait found on the X chromosome. Okay, and then the male, X little r, Y. And then so when he had these flies mate, we can draw a Punnett square. Let's go ahead and draw a Punnett square. Let's do X big R, X big R, X little r, Y. Okay. Oops, I made a mistake. It's supposed to be X big R, Y. X big R, Y. Okay. He found that both females and males had red eyes, right? I don't see any flies in the F1 generation with wide eyes, but all the females are the same. All the females are heterozygous. They are carriers for the white-eyed mutant gene. They are carriers. And the males are all the same too. They have the red eye X chromosome. And so he had them mate. So let's go ahead and draw a Punnett square for them. X big R, right? The females are carriers, are heterozygous. The males are red eyes. X big R, X big R, X big R, X little r. So the females, half the females are homozygous wild type. The other half are heterozygous or carriers. And then when we look at the males, half the males have red eyes. And then the other half of males have white eyes, and that's exactly what he saw. He saw often, whenever he saw a wide-eyed fly, it tended to be male, which was the X little RY. Okay, this guy right over here. So this was his experiment. This is how he figured out that the gene for eye color, red versus white, is sex-linked, and that it is not autosomal. Okay, because if you were to do a Punnett square, if it was autosomal, you would get different results. Okay, but he discovered that most of the time he found the wide-eyed on the males. So next topic is linked genes versus unlinked genes. What does linked genes mean? First of all, what is unlinked? Unlinked means that you have two genes that are found on different chromosomes. Okay, the two genes is sorted into gametes independently of each other. So when you do like a dihybrid cross, we're dealing with two traits at the same time, you're going to end up with 4 different kinds of gametes. And when you end up with 4 different kinds of gametes, you can do the 4x4 large dihybrid cross. You're going to end up with a 9:3 to 3:1 ratio in the F2 generation. Okay, in the F2 generation. Whereas with linked chromosomes, you're going to start with genes that are on the same the same chromosome. So these genes are on the same chromosome. So they do not assort into gametes independently of each other. And you're going to end up with unexpected phenotypic ratios. Okay? And Thomas Morgan and his, I guess you could say, student, okay, Sturtevant was his name. I forget his first name. Andrew Sturtevant? I forget his first name. But they worked together. Sturtevant was Morgan's student, and together they did these linked genes experiments. So what did Morgan and Sturtevant study? They studied specifically two genes in fruit flies. Body color, okay? And there were two kinds of body color. There was the gray versus the black, okay? And gray was dominant to black. So we're going to use the gene for gray, big G, and the gene or the letter for black is going to be little g, because black is recessive to gray. And then wing shape, you have normal wings versus vestigial. Okay? You know, vestigial wings are basically small, unusable wings, okay? They're like small, wrinkled wings where the flies cannot even use them. They can't fly with vestigial wings, and it's a mutation, okay? So black and vestigial, those are the mutant versions, whereas gray and normal wings are going to be the wild type, okay? So little n will be the symbol for vestigial wings. All right, so let me just attempt to draw like a fruit fly. Oh, I'm not the best drawer. So there you have normal wings, whereas vestigial wings, so there's the body, are tiny, tiny wrinkled wings, and they're unable to fly. Okay, that would be a mutation called vestigial. So what did Morgan do? Okay, he took a P generation, and the P generation, they were big G, big N flies. Okay, so they had gray bodies, normal wings, and he had them mate with black-bodied flies with vestigial wings. And when you do the Punnett square, right, you're going to end up within the F1 generation, they're going to be all heterozygous. They're all going to be heterozygous, okay? So then the F1 generation are going to be 100% big G, big N, little n. Okay, and then what he did next was he took a fruit fly that was heterozygous, and he had them mate with ones with black bodies and vestigial wings to produce the F2 generation. So let's go ahead and draw a Punnett square. Okay, little g, and then ends. Let's do the ends together. Okay. And big N, little n, little n, and little n. And when you look at this is a dihybrid cross, right? When you look at the results, he expected to get like 25% gray body with normal wing, 25% with black body, normal wing, 25% gray body, vestigial wings, 25% black body, and vestigial wings. Okay, so if you were to do the crosses here, you would see that it is 25. He expected 25% of the 4 different kinds of flies: gray and normal wing, and then gray and vestigial wing, and then black body with normal wings, black body with vestigial wings. That's what he expected. Okay, in the F2 generation, that's what he would have expected. Maybe I should do the crosses. Okay, so then gray and normal. Okay, when you do gray and normal, that's going to be 1/2 times, that's these guys, 1/2 times 1/2, right? That gives me 1/4, or 25%. And then gray and vestigial. And I'm doing this just to show you what he expected, but that's not what he actually saw, okay? And then so gray and vestigial, and then black, normal, is also going to be 1/2 times 1/2 equals 1/4, and then black, vestigial is also 1/2 times 1/2 is equal to 1/4. Okay, so that's what he expected, but that's not what he got. So here on the next slide is what he got. He had expected a quarter to be the wild type, gray body, normal wings, black vestigial, a quarter black vestigial, a quarter gray vestigial, a quarter black normal. But that's not what he saw. Instead, he saw the majority of flies that look like the parents, and he called them the parental phenotypes. So 965, 944, 965 looked like this parent, and then 944 looked like this parent, and then there were just a small number of what he called the recombinant phenotypes. So recombinant phenotype, what does that mean? That means that we're talking about gray vestigial. You know, it's not like the parents, okay? Black normal wing, it's not like the parents. So the recombination, okay? And so from this experiment, they were able to figure out that, so maybe these genes for body color and wing shape, maybe they're linked. Maybe they do not independently assort into gametes. If they were on separate chromosomes, then they would independently assort. But since you see most of the phenotypes to be like the parents, maybe they're on the same genes. And then the recombinant types are due to crossing over, okay? So again, he used these strange letterings, okay, like B plus, VG plus. Personally, I just use, you know, the GG for gray, big G for gray, big N for normal, okay? And then, actually, you know what? This is the heterozygote type, so let me go ahead and erase that, because we're talking about the F1, okay? And remember, the F1 were heterozygotes. So let me go ahead and change my pen. So for the F1 hybrids, they were big G, big N, little n, and he had them mate with a black-bodied vestigial wing, so little n. And so because the genes are linked, so maybe I can draw it like this instead. So you have big G, and maybe big N, and then you have little g, little n, okay? So that's the homologous pair that's in this fly. Okay, these are the homologous pairs. Then you can have some crossing over take place. After crossing over in meiosis, then you're going to end up with sister chromatids that are going to be recombined or recombinant. So this is going to be big G. Actually, I want to, yeah, okay, I can do that. I want to change that to little g, because it makes sense that the tip would cross over. Okay, so let me go ahead and erase that and change that to a little g. So there's a little g and big N, and then over here I have a big G and a little n. Okay, so crossing over had taken place during meiosis. And that's how you end up with the recombinant. That's how you end up with gray bodies, vestigial wings. A small portion of the flies did come out recombinant due to crossing over. Okay, but the majority of the flies looked like the parents, and he called them the parental type offspring. And then there was a small portion that was the recombinant type. And so what did Sturtevant and Morgan do? They took the numbers of the recombinant offspring. So they added the numbers together, 206 plus 185, that equals 391 recombinants. They divided by the total number of offspring. So they divided by 965 plus 944 plus 206 plus 185. And then they got this frequency of 0.17. They call that the recombinant frequency or recombination frequency, and then they multiplied it by 100%. to give you 17%. Okay? And that 17%, they decided that, maybe the amount of recombination or the recombinant frequency correlates with how far the linked genes are from each other on the chromosome. Okay? So let me share a slide, like a whiteboard, a white screen. So what Sturtevant and what Morgan were thinking was that if you have, let me go ahead and draw here. So here's my homologous chromosomes, right? So if they're, let's do A, big A, and maybe big B, and then these could be like little a's, and then these could be little b's. The farther the chromosomes are from each other, there's a greater chance of recombination. There's a greater chance of crossing over. Okay, so then you're going to end up with like big A, big B, and then this one's going to have like a little a and maybe a big B. And then this sister chromatid that crossed over would now have the big A, and then there's a little b there, and then a little a and a little b. So you guys, those two sister chromatids, right over here, maybe let me change the pen color so it looks a little bit more obvious, okay? So, you know, this sister chromatid, those are the chromatids that are going to give you the recombinant offspring, okay? The recombinant offspring. So what they were thinking is that the farther apart they are, there's probably going to be a better chance of recombination. Whereas if they were closer together, so versus, let's draw it over here. If the linked genes are close together, so big A, big B, big A, big B, little a, little b, little a, little b, the likelihood of them crossing over and switching portions of DNA is less. Okay, the likelihood of that happening is less. So the recombination frequency is going to be pretty small. So the distance between the two genes, the linked genes, is going to be shorter. Okay, they're not going to be very far from each other. So that's what Morgan and Sturtevant found, was that when you have when you figure out the recombination frequency, that correlates to how far the linked genes are from each other, the location of those chromosomes. Okay, so going here, let me just see here first. So again, from their experiment, very few were gray and vestigial, and a few black and normal. Those are the recombinant types. He attributed these few recombinant types as a result of crossing over. Okay, so yeah, this is what I was trying to explain on the previous slide, how you can have crossing over and then producing these sister chromatids with that do not represent or don't look like the parents. Okay, it's a recombining of like gray body with vestigial wings or black body with normal wings. And from that, he was able to, or Sturtevant and Morgan, they came up with this idea of making up a chromosome map. So the further away the chromosomes, if they're linked, the further away the genes are from each other on a chromosome, there's going to be a higher frequency of recombination. Okay, so here is a chromosome map of some of the genes that are found on one of the chromosomes. Specifically, it looks like chromosome number 2, pair number 2 of a fruit fly. Okay. So how did they come up with the chromosome map? Okay, the recombination frequency. So between B, so this was the gray body and normal wing, the distance between them, it was like 17%. Remember that number? that was the recombination frequency. So they said that, oh, it's about 17 map units apart, okay? And then between gray body and then there's this other eye color called cinnabar, it was a 9% recombination frequency. And between cinnabar and normal wings, it was about 9.5 recombination frequency, all right? So the closer the two genes are on a chromosome, the less likely they are to cross over. And you can use the percentage of recombinant phenotypes to determine the distance between the genes. And usually 1% crossover is equal to 1 map unit. And these are just, they're not exact locations, they're just approximations, okay? In a black-bodied purple, for example, black-bodied purple eye cross, 6% of the offspring are recombinants, and so that means, it means 6 map units apart, okay? And results can make a chromosome map like you guys saw on a previous slide. So here's a sample gene mapping problem. Use the following crossover frequencies to create a genetic map for genes A, B, C, and D. And I'll give you guys, after this one, an actual problem on how to figure out frequency, okay? Recombinant frequency. So step number 1, when you see these genes, and we're talking about genes A, B, C, all the way to D, I would take a look at the distance between the genes, or the gene frequency, crossover frequency. Find the one with the largest number. That means A and D are the furthest apart. So let's go ahead and just like draw. So there's gene A, there is gene D, and the distance between them is 35. So maybe we can go like this. I think that would be the easiest thing. So 35. So we did that one. Next one is between A and C, which is 25. I mean, we could put C here and then make that distance 25, right? Or could we put C here on this side? Could we put C on this side and make that 25? Well, let's take a look. Well, so that's between A and C. A and C is 25. But then take a look, between D and C is 10. Okay, so between D and C is 10. If we put C over here and make this 10 and this 25, that works out, right? Between A and C is 25 and between D and C is 10, okay? All right, next one, we have to map out where B is. Where would B be? B. We could put, between B and D, it's 14, so we could put B on this side. Or we could put B on this side. But here's the thing, between B and C, the distance is 24, right? It's 24. So how is that going to work out? Well, you know what? I think we're going to have to put B on this side. And I've kind of run out of space. So it's not going to look like, it's not to scale. Okay, let's try this out. If we put B here, between B and D, it's 14. Okay, so that's 14. And then between B and C, it's 24. So that does work out. Okay, 24, right? It's 10 plus 14 is 24. Between B and C is 24. So that works out. All right, so that's how you can map out a chromosome based on recombination frequency or crossover frequency. So let's go ahead and do a sample problem on how to figure out these frequencies. Okay. I actually had to pause the video there because I didn't like the practice problem that was in the original PowerPoint, so I've modified the PowerPoint with a different problem that makes a little bit more sense, okay? So this is a practice problem for determining the recombination frequency, and from there we can determine just how far apart, how many map units apart these, if these genes are linked, how far apart they are on the chromosome, okay? So here we have a wild-type fruit fly, heterozygous for gray body color and normal wings, was mated with a black fly with vestigial wings, okay? So from here, let's go ahead and just write the genotypes out on this slide. So heterozygous for gray body color. So it's heterozygous, so big G, little g, and then normal wings, big N, little n. Okay, that's that fruit fly. Was mated with a black fly with vestigial wings. So then the other parent is little g, little n, little n. And then the offspring had the following phenotypic distribution. So these are the parents, and then these are the kids. 778 were the wild type, meaning they had gray body and normal wings. And then black vestigial, 785, like this parent. So you guys, the first two number sets are the parental types. These are the parental types. So P, I'm going to write P for parental because they resemble the parents, having wild type phenotype or having the mutant phenotype, which is black body and vestigial wings. And then here you have 158 and 162. These are the recombinant types. They do not resemble their parents, rather it's a mix of the parents. Okay, so these are the recombinant types. You have like black with normal wings and then gray body with vestigial wings. What is the expected phenotype? So we're answering this question now. What is the expected phenotype if the genes have segregated randomly? So if they were on separate genes, if the genes were on separate chromosomes, then we would expect, like Morgan and Sturtevant did, they would have expected this 25% wild type, 25% black vestigial, 25% black normal, and 25% gray vestigial. So you would have expected this 25% to 25% to 25% ratio, or 1 to 1 to 1 to 1 ratio, okay? But we don't see that here. You can look at the numbers and you can automatically see that it's not 25%, 25%, 25%. It's not equal. It's not this 1 to 1 to 1 to 1 ratio. So are the genes linked? Yes, we would say that they are linked. Yes, okay, and you would explain why. Because we do not see this 1 to 1 to 1 to 1 ratio. We don't see 25% of each type represented. Instead, we see the majority of the flies resemble the parents, and then a small number resemble the recombinant type. So that tells us that the genes are linked. What is the recombination frequency between these genes for body color and wing type? So we just have to take these two numbers, you add them together, the recombinant types, you add them together, and then you divide it by the total number of flies. So it's 158 plus 162. 158 plus 162 divided by the total number of flies, which I added up earlier, 1,880 flies, and that gives me 0.17. And then you need to multiply that by 100% to give you 17%. That is the recombination frequency, and that is equal to the map units apart. So when you look at, this is an estimation, okay? So these recombination frequencies are estimations. They're not exact locations because when you look at a chromosome map, it's based on recombination frequencies, and you're kind of finding the general area or distance that the genes are from each other, okay? So it's about 17 map units apart between the body color gene, either big G, little g, or the wing shape, big N or little n, okay? That's the distance. Okay, so that's how you figure out, that's how scientists will map out a chromosome, is they'll take all these different genes that they know that are linked, and then they'll figure out the recombination frequency, which is equal to the crossing over frequency. The closer the genes are, recombination frequency is going to be small, right? Crossing over frequency is going to be small. But the further apart they are, crossing over is going to be happening more frequently. So recombination frequency is a lot bigger, and so they're going to be further apart. I hope that makes sense. Okay, next topic, what are the two alterations of chromosome number or structure that cause genetic disorders? Number 1 and 2 is, number 1 is non-disjunction. So we're going to talk about that first, and then we'll talk about, the last thing we'll talk about is alterations of chromosome structure. What's non-disjunction? Non-disjunction is the alteration of chromosome number, and that will happen in meiosis. That can happen in meiosis. There's going to be change in chromosome number in the gametes. So if you look at the gametes down here, okay, if you look at the gametes down here, there can be a mistake in meiosis 1 or meiosis 2 that's going to give you a haploid number of chromosomes with 1 extra chromosome or 1 less chromosome. Okay, so this is the first, I guess you could say, first instance where non-disjunction is going to happen. So remember in meiosis 1, the law of segregation where the homologous pairs separate? Over here, let's say that the homologous pairs, they do not separate. Oops, sorry. That's really loud. I'm going to just lower the volume so it's not too loud when you guys watch the video. So let's say that the homologous pairs fail to separate. And if they fail to separate, then this cell right here is going to get 2 extra sister chromatids. See these sister chromatids? They went into the wrong cell. So you're going to end up with half the gametes with an extra sister chromatid. So it's going to be a haploid number plus an extra chromosome. Haploid number plus an extra chromosome. And then this cell is now missing that chromosome. Now it's missing sister chromatid. So it's going to be a haploid number minus 1. Haploid number minus 1. So all the gametes here are now going to have the wrong number of chromosomes when there's non-disjunction happening in meiosis 1. A second way that non-disjunction happens is over here in meiosis 2. Let's say the homologous pairs, they do correctly separate. By the law of segregation, they do separate and segregate correctly. But then in meiosis 2, remember in meiosis 2 where the sister chromatids separate in anaphase 2, let's say that there's non-disjunction that happens here in meiosis 2. In anaphase 2, the sister chromatids do not separate. Therefore, this cell is going to end up with an extra chromatid. This cell is going to be missing an extra chromatid. And then these are okay. These gametes are fine because meiosis 1 was fine, and then meiosis 2 was fine. But for meiosis 2 in this cell, the sister chromatids didn't separate. So you're going to end up with 2 gametes that have the wrong number. 2 gametes that are fine. Okay, so I guess the worst case scenario for non-disjunction would be when there's a mistake in meiosis 2. That would give you 4 gametes with the wrong number, whereas if there's non-disjunction happening in meiosis 2, half the gametes have the wrong number. So in the first case, where you have non-disjunction happening in meiosis 1, all the gametes have the incorrect number, whereas with non-disjunction in meiosis 2, half are going to have the wrong number. So non-disjunction is going to result in gametes with the wrong number, and when fertilization takes place with the wrong number, you're going to end up with a zygote with an extra copy, or maybe a zygote that's missing a chromosome. Trisomy is where there's an extra copy of a chromosome. So instead of having 2, it has 3 of that chromosome. So here is that, maybe let's say egg. So let's say this is the egg where there was a mistake in meiosis due to non-disjunction. There's an extra chromosome. So then you're going to end up, here's that extra chromosome plus 1. That's called trisomy, because instead of 2, you now have 3 copies of that chromosome. And then let's say that you have the egg or gamete that's missing a chromosome. Then you're going to only have, instead of having 2, you're missing a chromosome, so you only have 1 copy, and that's called monosomy. Monosomy, mono means 1, tri means 3. Okay, so these here, these are gametes with an extra chromosome, or that's missing a chromosome. That's due to non-disjunction. So there's trisomy, where you have extra copy, so now you have 3 copies instead of 2. Monosomy, where you have 1 copy of a chromosome instead of 2. So a really good example of trisomy is trisomy 21 or Down syndrome. So here, if you take a look at the karyotype of someone with Down syndrome, chromosome number 21, there is 3 copies. Everything about the karyotype is completely normal. This is a female karyotype. Everything about this karyotype is normal. There's 2 of every chromosome except chromosome 21, there's 3. So this is a trisomy 21 because it has 2 sets of chromosomes, except chromosome number 21, there's an extra chromosome. So this person has 2 sets of chromosomes plus 1 chromosome. It's going to be 46 chromosomes plus that 1, so total of 47 chromosomes. So this person, if you count all the chromosomes, they have 47 chromosomes, not 46, 47. So trisomy 21 is Down syndrome, 3 copies of chromosome 21, and it's fairly common in the United States, around the world. It's 1 in 700 children are born with Down syndrome. Chromosome 21 is a small chromosome, but having those extra genes, that extra DNA has severe effects. Okay, you can have, different range of ability, intelligence, or ability. Some people with Down syndrome need, it's so severe that they need 24-hour care, whereas some people with Down syndrome, they can cook for themselves. They're pretty independent, okay? Frequency of Down syndrome correlates with the age of the mother. Remember how we were talking about in oogenesis, how oogenesis doesn't complete until fertilization takes place. So when a woman is older, her eggs are older, and so there's going to be a greater chance of mistakes in meiosis. And that's why you see a higher frequency of Down syndrome the older the mother is. And then there's also sex chromosome abnormalities. Human development is more tolerant of the wrong numbers in sex chromosomes. So I'll give you guys some examples of that. But produces a variety of distinct syndromes in humans. So for example, XXY, this person has an extra X chromosome, and that's called Klinefelter syndrome. Because there's a Y, they are considered a genetic male. You guys, in order to be a genetic male, you need to have that Y, and Klinefelter's is where you have that extra X. So let me just move this up here so you guys can see. Let's say that there was nondisjunction happening in egg formation. So this egg has an extra X chromosome, and together, okay, with the Y chromosome, when you do this Punnett square, together you're going to end up with a male with Klinefelter syndrome, that extra X. And we're going to be talking about some of the details of Klinefelter syndrome, okay? And then there's also the extra X meeting up with the X chromosome from the sperm. You get a female with triple X syndrome, and we're going to be talking about that as well. This egg didn't receive that X chromosome, so then she would have monosomy, right? Monosomy. It's missing an X chromosome. That's called Turner syndrome. And then if this egg doesn't have an X chromosome, it meets up with the Y chromosome in the sperm, there's no X chromosome, then it's non-viable, meaning that that zygote will be miscarried. It will never develop. The X chromosome has hundreds, maybe even thousands of genes on it that are absolutely important and necessary for development. Okay? So if there's no X, it's not going to survive. Oh, and then there could be mistakes there could be non-disjunction happening with sperm formation, okay? And so actually in this case, I'm thinking about, you're going to end up with 2 Y chromosomes, okay? 2 Y chromosomes. And so that's going to lead to something called Jacob syndrome or XYY. And we'll talk about how that happens, or what are the results of Jacob syndrome. So first of all, Klinefelter syndrome in the male, it's XXY, and you can see that here in the karyotype. See how there's supposed to be 2 sex chromosomes? Well, this person has 3 sex chromosomes. 2 of every chromosome except the sex chromosome, there's going to be an extra X. So this is a type of trisomy. Okay, trisomy. Trisomy, it's a type of trisomy because they have 3 sex chromosomes. 1 in every 2,000 live births. Males will have sex organs, but oftentimes they tend to be pretty, underdeveloped, so they might be sterile. And then they might exhibit some feminine characteristics like breast development, lack of facial hair. They tend to be pretty tall in height and completely normal in terms of intelligence, average intelligence. And then Jacob's syndrome is where you have the 2 Ys, okay? And you know what? I was thinking, like, how does that happen? Because, in a normal male, you have XY, and then during, the meiotic process, the law of segregation, you're going to have them separate. Perhaps, maybe, and I'll have to look this up to verify, maybe cytokinesis cytokinesis failed to happen when you have the sister chromatids of the Y. Maybe the sister chromatids failed to separate, or maybe cytokinesis didn't occur, in that last stage of meiosis. So you have the extra Y, and this occurs 1 in 1,000 live male births. Extra Y chromosome, trisomy, right? This is again a type of trisomy. Slightly taller than average, more active, normal intelligence. They might have slight learning disabilities, maybe ADD or ADHD, delayed emotional maturity, normal sexual development. So that extra Y chromosome, you have extra genes, and maybe some of those genes are being overexpressed. They're making lots of the proteins that are associated with the Y chromosome, and that makes them more active, okay? And then trisomy X is where you have a female with 3 X chromosomes, 1 in every 2,000 live births. However, females are healthy. Why? Because I hope you guys remember, we talked about how in a cell, in a female, one of the X chromosomes is inactivated. We call that inactivated X chromosome the bar body. So no matter what, in all of the cells in a female, only 1 X chromosome is active. Only 1 X chromosome is active, okay? And then the other 2 X chromosomes will become bar bodies. So if you take a look here with the 3 X's, 2 of them are going to become bar bodies, and only 1 will be active, okay? All right, there's something called Turner syndrome. Turner syndrome is monosomy X, 1 in every 5,000 births, varied degrees of effects. Okay, webbed neck, short stature, and they're usually sterile due to rudimentary ovaries, meaning that the ovaries have not fully developed. Okay, so they're usually going to be sterile. I actually did have a student with Turner syndrome, and she was very open about it. This was years ago, probably over 10 years ago. And she had the classic, Turner syndrome phenotypes. And she knew that she was sterile, so she shared with the class too that she was planning on adopting as many kids as she could. So she wanted a big family through adoption. So here's the thing, with humans, having those extra chromosomes is usually problematic, but with plants, having extra sets of chromosomes, so this is different. Polyploidy is having extra sets, so like not just 2N, but like having 3N, triploidy, having 4 sets, tetraploidy, having 5 sets, 6 sets. So we're not talking about what we were talking about with nondisjunction, how it's like 2N plus 1 chromosome, or 2N minus 1 chromosome. That's not what we're talking about. When we say polyploidy, we're talking about sets of chromosomes. So let's say that, and I'm totally making this up, but let's say in a strawberry, a normal strawberry, you would have, 16, okay, 2N is equal to 16 chromosomes, but then 3N, so it's 8 plus 8, right? So one set would have 8 chromosomes, so it's 2 plus 2, one set plus one set, or 2N equals 16 chromosomes total. But 3N is 8 plus 8 plus 8, that would give you 24 chromosomes. So you know what I'm saying? We're like, when we go with polyploidy, we're actually adding a whole set of chromosomes. And with plants, polyploid is very common. In fact, the more sets of chromosomes that they have, they tend to yield larger fruits. Like with a normal strawberry, it would be pretty small in size. But when you make them into a polyploid by in agricultural developments, you can modify plants to be polyploid, and so you would end up with larger size strawberries. I think in your textbook it said that strawberries have, I think, 6 sets of chromosomes, or was that the wheat? I forget. But there's strawberries that you get at the supermarket are polyploid, and then also wheat is polyploid as well. Banana, yeah, in your textbook, I remember this. Bananas are triploid. They have 3 sets of chromosomes. Okay, next and the last thing that we're going to talk about are alterations of chromosome structure. So these are mistakes in chromosome structure. There's problems that could be due to errors in replication. As DNA replicates, and do you guys remember when DNA replicates? Like what stage of the cell cycle do they replicate? And I'm hoping you guys are all saying the S phase, okay? During the S phase of the cell cycle, perhaps there's a mistake in DNA replication. Maybe there's a deletion, a part of the DNA that is missing. That's deletion. So in this example, and let me just move my head out of the way. In this example, okay, these are the sections of the chromosomes, A, B, C, D, E, F, G, H sections. Let's say during replication, the D was not copied. It was deleted. And so you have A, B, C, E, F, G, H, okay? And in fact, there is a genetic disorder called Cri-du-Chat. I think that's how you say it. It's a French term. Let me just write it here. Cri-du-Chat or chat. I don't know how you say that in French, okay? But it means cry of the cat, where chromosome number 5 has a deletion. So let's say this is chromosome number 5, and there's a part of it that's gone. It's deleted, okay? There's part like up here is like now gone. And it causes some mental disabilities in terms of intelligence, and then also they tend to have microcephaly, so a small head. But it's called cry of the cat because when they're babies, when they cry, they sound like a cat meowing. Very strange initial diagnosis of the Cri-du-Chat is that the babies would cry like a cat. Okay, so that's an example of a deletion. So loss of a chromosomal segment. And then duplication is in the S phase. Maybe a part of the chromosome will be duplicated. You have 2 copies of. So a segment is copied twice. So you have A, B, C, D, E, F, G, H. But then you have here B and C being copied twice. Okay? And then there's a repeat in the segment. And then there's an error maybe in crossing over. Inversion. So instead of A, B, C, D, E, F, G, H, it's A, D, C, B. So maybe during crossing over, when there is exchanges in the segments, instead of going in as B, C, D orientation, it's gone in backwards, and that's called inversion. And then you also have translocation, where you have a piece of the chromosome not attaching, it's not reattaching to the appropriate chromosome, it's going to attach somewhere else, okay? So that's due to crossing over, and that's called translocation. So see how you have C, D, E, F, G, H, and this AB went somewhere else, and then an M and O from another chromosome attached to it, okay? So it's the movement of segment from one chromosome to another chromosome, the wrong chromosome. I want you guys to think about, like, which of these 4 alterations of chromosome structure, which of these 4 is the most damaging? Which of these is the most damaging? Deletion, duplication, inversion, or translocation? Which would be the most damaging? Okay, hopefully you said deletion. Deletion is the most damaging because you're actually losing genes, whereas the other cases, you're not losing genes. The genes are still there, the segments of the DNA are still there, but in deletion, you're actually losing DNA. Okay? So, if there's a chromosomal alteration, or maybe non-disjunction, you can get tested, and this is kind of an old way of testing. Now there are more updated versions of genetic testing, simple blood tests that can tell you, looking at the karyotype of the fetus, you can see if there's any chromosomal abnormalities, okay? But an amniocentesis is pretty, it's an old method, but they still do it, okay? What is an amniocentesis? They basically want to get fetal cells out of the amniotic fluid. So when a woman is pregnant, they'll take this very large needle, hypodermic syringe needle, and they'll draw up some amniotic fluid, and that amniotic fluid has some fetal cells floating in it, and then they'll grab those cells and they'll take a look at the chromosomes and analyze the chromosomes in a karyotype, and the karyotype can see if there's like 3 chromosome 21s, then they know that the fetus has Down syndrome. If it's missing a piece of chromosome 5, that it has Cri-du-Chat, okay? So an amniocentesis is usually done in the 2nd trimester. There's other tests that can be done much earlier, okay? I had my son later on in age, so I had him in my late 30s, and so the doctor was like, oh, maybe we can do an amniocentesis. I was like, no, I don't want to do amniocentesis because there is a little bit of a risk of miscarrying the child, because if you think about it, you're putting a big needle in there. You might puncture the amniotic sac and then, cause early labor. So I didn't want to risk it. I mean, I waited years for him. I'm not going to lose him due to some genetic testing. So I didn't do that. And also, I don't really like needles, so I didn't like the idea of sticking a huge, long needle through the uterine lining into the amniotic sac. It just wasn't feasible for me. So again, that's what a karyotype looks like, right? And I'm going to skip this section on the pedigrees because in your textbook, actually chapter 15, it does go over pedigrees, which we went over in chapter 14, so we're going to skip that. Okay, but with pedigrees can be helpful with genetic counseling. Pedigrees can help us understand the past and predict the future. Genetic counselors can look at pedigrees and figure out what is the probability that if you might have a recessive genetic disorder that goes through your family, what's the probability that you're going to pass it on to your kids, okay? Here are some benign to deadly diseases like albinism. That's pretty benign, right? Cystic fibrosis could be deadly, as is Tay-Sachs. Sickle cell anemia, however, is very treatable. PKU would be a deadly disease. Okay, so these are some common recessive genetic traits, okay, that can be passed down from one generation to another that you can see in a pedigree. So recessive diseases, they're recessive because of allele coding for a malfunctioning protein is not going to show up in carriers. So people that are heterozygotes, they carry the recessive trait, but they're going to show a normal phenotype because it's dominant. Okay? So this is like the way that it would work. Heterozygotes as carriers of a recessive trait. So let's say you have 2 parents that are heterozygous. They both carry that trait. Then there's a 25% chance of having a homozygous normal offspring, 50% chance of producing a carrier, and then a 25% chance of having a child with the disease. Okay? So if you know that in your family, let's say that you know that you're a carrier, and then your partner is a carrier, and then there's a 25% chance of having offspring with the disease, A genetic counselor would counsel you on this and tell you the probability and the risks of it, and then you could decide whether or not to take the risk. So that's something that, I mean, would you take the risk? If you knew that there's a 25% chance of having a child maybe with cystic fibrosis or Tay-Sachs disease, would you take the risk? That's something that is a very personal decision that you would have to make with your partner, with the help of a genetic counselor. Here's that example of a recessive disease called cystic fibrosis. It's primarily found in Caucasians of European descent. It's 1 in 2,500 births. And in fact, 1 in 25 Caucasians are carriers for cystic fibrosis. The normal dominant allele, it codes for a chlorine ion channel protein. Okay, but someone with cystic fibrosis, they have a chlorine ion channel protein that is misshapen. Okay, it's either defective or it's absent, and therefore they can't transport chlorine across the cell membrane. And because they can't do that, it causes mucus to build up around cells, and specifically it will build up around digestive glands like the pancreas. It will build up in the lungs, so it's very difficult for someone with cystic fibrosis. It would be very difficult to breathe, okay? And without treatment, children can die before age 5, but with treatment, they can live past their late 20s, okay? And Tay-Sachs is a recessive trait. Those of you that saw the movie, oh no, I didn't show it to my students last year, I don't think, Lorenzo's Oil. Actually, we probably saw that movie, Lorenzo's Oil. I believe we, really sad movie, but it is very similar to Tay-Sachs, where there is a non-functional enzyme in the brain, and therefore there's a buildup of lipids in the brain cells that causes brain cells to be destroyed, okay? And Tay-Sachs is primarily seen in Jews of Eastern European descent, like Ashkenazi Jews, and also Cajuns. People with Cajun background in Louisiana specifically, 1 in 3,600 births, and this genetic recessive disorder produces a enzyme that doesn't work, and this enzyme specifically breaks down lipids in the fat, so you end up building up lipids in the brain, which can destroy the brain. And symptoms begin few months after birth, such as seizures, blindness, muscular and mental degeneration. So those of you that saw Lorenzo's Oil, remember how Lorenzo, he started having, emotional fits, right? Started destroying things in his home and at school, and then he started having seizures. He couldn't see, he couldn't talk, etc. Remember that? Okay. So people with Tay-Sachs will usually die before the age of 5. Sickle cell anemia, you know what? Actually, it is a recessive trait, okay? However, it's not fully recessive. It's more, in a heterozygous person, it's actually codominant, okay? So this is I can't spell codominant. Codominant. It's not truly recessive, okay? It's codominant. So let's cross that out, okay? It's a codominant trait. Primarily found in Africans, 1 out of 400 African Americans have sickle cell anemia, and it's caused by substitution of a single amino acid in the hemoglobin protein. Hemoglobin protein, remember, it delivers oxygen to all of our cells, and so as a result, people with sickle cell anemia, their oxygen levels tend to be very low, and so cells can die, cells can be low on oxygen for cellular respiration, okay? And this is due to a substitution, okay, in the amino acid that makes up the hemoglobin protein. It causes the red blood cell to have this sickle shape. Sickle shape. It's shaped like a half moon, or a crescent moon, I should say. It's shaped like a crescent moon. And because it's shaped like a crescent moon, you can imagine that these red blood cells cannot move through the capillaries very smoothly. You can get blood clots, okay? And also it cannot deliver oxygen efficiently. So there is this pleiotropic effect. There are many, many different symptoms due to this one genetic mutation. Okay? Substitution of one amino acid in the polypeptide chain. It's going from a hydrophilic amino acid. So glutamine is a hydrophilic amino acid, but now it's being changed to valine, which is a hydrophobic amino acid. And do you guys remember when we talked about proteins and amino acids? The hydrophobic ones tend to, like, the hydrophobic amino acids, they tend to be move in the middle of the protein away from water, whereas the hydrophilic amino acids, they'll face towards the water. And so as a result, the hemoglobin in the red blood cells in a normal one would give the red blood cells this round shape, but in someone with sickle cell anemia, their hemoglobin has a very long chain shape, and so the red blood cells is going to have this sickle shape. It's going to be deformed. The cell will be deformed. And when it's deformed, the sickle cells, those red blood cells are not going to be efficient and functioning. They're going to cause clumping and clogging of blood vessels. And sometimes, you know, these sickle cell red blood cells, they can get stuck in the spleen and cause spleen damage. I mean, this is the pleiotropic effects of having sickle cell anemia. But here's the thing, what I mean by sickle cell being codominant is that someone with sickle cell, someone that's a carrier for sickle cell, they are heterozygous. So maybe we can say like big N, little n. Big N for like normal hemoglobin, or maybe I should use, yeah, I'll just use big N for normal hemoglobin. And then this would be the gene for the sickle cell. You know what? I should probably use 2 different letters, right? So let me use the 2 different letters. When we talk about codominance, we tend to use the 2 different letters. So I'm going to use N for normal, and then I'm going to use S for sickle. So someone that is heterozygous, normal, and sickle cell, half of their blood is normal shape. Their red blood cells are going to be normal shape. The other half are going to have a sickle shape. Okay? So 50% of the blood for someone that is heterozygous or carrier for sickle cell anemia, half of the red blood cells are normal shape, and then the other half, or 50%, are sickle shape. And this gives them an advantage. They have a literal advantage over people that are normal. So people that have normal red blood cells, 100% normal red blood cells, versus someone that has sickle, okay, 100% sickle shape. Somebody with heterozygous, they have a advantage because they're actually immune to malaria. Malaria is caused by a parasite. And these parasites, they spend some of their life living in healthy red blood cells. So you guys can see here, these are healthy red blood cells, but when they're infected with the parasite lives in these red blood cells, causing damage to red blood cells, and which causes that person to become very ill. Okay, so if someone has the gene for sickle cell anemia, they are heterozygous for sickle cell anemia, half of their blood is sickle shape, and these parasites cannot live in the sickle cells. So having 50% of the blood sickle shape, the malaria parasite cannot survive in that person's body because there's not enough healthy red blood cells for them to reproduce and live inside that person's body. So someone that is heterozygous for sickle cell anemia, they're free of malaria. They're immune to malaria, and that's called the heterozygote advantage. Okay? And then Huntington's disease is a dominant genetic disorder. Most genetic disorders are recessive. This is a dominant one. I guess the most important part here is that in Huntington's disease, there's a buildup of this protein in the brain, causing brain cells to die. It usually starts with memory loss and then muscle tremors. And here's the weird thing about Huntington's disease. It starts at age 30 to 50, so later on in life. If you knew that Huntington's disease was in your family and you knew that it's a dominant trait, I don't know. I mean, I don't know, like, it's a personal decision, but I don't know that I would want to pass that on to my kids. So I don't know if I would plan on having biological children, because it starts later on in life too, right? Testing, would you want to know? I think I would want to know so that I would plan for the future and maybe plan not to have biological children. Okay, and I already showed you guys this one, so we don't have to go over hemophilia, the pedigree of the royal hemophilia pedigree, but we are going to go over how hemophilia is a sex-linked recessive trait. So this was Queen Victoria, who was a carrier, and she, her husband was a, normal, no hemophilia man, and then there was a, out of all the girls, there was a 50% chance of having a girl that was a carrier that could pass that down to her kids. And then out of the boys, when you just look at the boys, there's a 50-50 chance of having a son with hemophilia. But out of all the kids, there's a 25% chance of having a child with hemophilia. But when you just look at the boys only, it's a 50% chance of having a son with hemophilia, okay? And that is because by the law of segregation, right, she could either give a healthy X versus a hemophilia X, okay? Whereas her husband did not carry the hemophilia gene. He had a normal X or a Y. So all males get their Y chromosome from their dad and their X chromosome from their mom. All right, let's try this problem. A couple who are both carriers of a gene for cystic fibrosis have two children who have cystic fibrosis. What is the probability that their next child will have cystic fibrosis? I mean, if you think about it, you're like, oh, they have 2 kids with cystic fibrosis. Their next one probably won't have cystic fibrosis, but that's not true, right? You need to do a Punnett square that will show you. So we'll do big N, little n. So they are carriers for cystic fibrosis. They do not have cystic fibrosis, so they're gonna carry it, but they don't show it because cystic fibrosis, remember, is a recessive trait. So big N, big N, big N, little n, big N, little n, little n, little n. So there's a 25% chance that they're going to have another child with cystic fibrosis. Okay, so their first 2 children, there was always this 25% chance, and I guess they took the risk. And then this is the same problem. What is the probability that their next child will be phenotypically normal? Going back to this Punnett square, there was a 75% chance of having normal phenotype. A woman and her spouse both show the normal phenotype for pigmentation, but both had one parent who was albino. Albinism is an autosomal recessive trait. If their first 2 children have normal pigmentation, what is the probability that their third child will be albino? So it says here that the woman and her spouse, they both have one parent who was albino, and albinism is a recessive trait. That means that both of their parents they both had a parent that was little, you know what, I'm going to use a different letter because normal is the dominant trait. So whenever I do these type of problems, I like using big N for normal and then little n for the recessive genetic disorder, okay? So that means that their parents they had at least one parent that was little n, and these parents, the woman and her spouse, are both normal. So that means that they must be carriers, right? They must be carriers. So I'm just going to write it like this then. Big N, little n. They must be carriers because one of their parents were albino. So their first 2 children have normal pigmentation. What is the probability that their third child will be albino? 25% chance. And then there's a 75% chance of having another kid with normal pigmentation. But there's always going to be a 25% chance or 1/4 chance of having a kid with albinism. Okay? think that might be it. Yes, it is it. Okay. All right, everybody. I hope that was helpful, and I don't think that this video was as long, but I hope that it was helpful. Okay. Bye.